So welcome back everyone to episode 9. So today we will be discussing about the anti-D typing. So we will discuss the basics starting from the slide technique on how do we do for the D typing, then moving on to the tube technique and then by the microplate technique. The next video we will be discussing about the identification of weak D and the various techniques which are used for it. So I have Dr. Anand today who will be taking you through all the steps for identification and typing of D antigen. Over to you, Dr. Anand.
These are the requirements we need for testing anti-D in slide. First, we need a monoclonal anti-D reagent. We also need a monoclonal control that is common for both slide, tube and microplate testing. Now we also have cleaned and labeled slides for anti-D and control. On proceeding with testing, first we will be placing one drop of monoclonal anti-D reagent to the labeled slide and another drop of monoclonal control to the labeled slide. Now we are adding approximately prepared 40 to 50% cell suspension into the slides. We will be adding one drop each to the monoclonal anti-D reagent and one drop to the control slide also. Here we are mixing the reagent slowly with a clean glass slides in a circular fashion. We are doing the same on both the slides for about 20 millimeter diameter. After that, we will be tilting the slides to observe for an agglutination. On the slide of anti-D, we can see a clear macroscopic agglutination, whereas on the control slide, there is no visible agglutination. Now coming to testing on tube method for anti-D. Here we have two labeled tubes of anti-D and control. Adding one drop of monoclonal anti-D reagent to the labeled anti-D tube. Another one drop of monoclonal control to the corresponding labeled tube. And we also have the 3% red cell suspension we have prepared from the sample. Here we are adding one drop of the prepared 3% suspension to the tube of anti-D and the same one drop of suspension to the control tube also. Now we are mixing the added reagents on both tubes and transferring them to the centrifuge. We must ensure that the weight is balanced on the opposite sides, closing the centrifuge, and the centrifugation has to be done as per the manufacturer's instructions. Now coming to the result analysis after centrifugation. On gently suspending the tubes, we can see that a clear 4 plus visible agglutination on the tube of anti-D, whereas on the control tube, there was no visible agglutinations. Here, we will be doing the anti-D testing on microplate using SPRC technique. For that, we will need the barcode label EDTA sample, which will be inserted on the rack. The rack will be selected accordingly on the machine, and it will be inserted into the machine on the designated slot. This is a microplate that contains multiple wells for testing. First, we have to place a microplate into the transport frame. The transport frame will be then placed on the loading tower on the designated slots. After loading the sample and giving specific instruction for running the test, these are the final results that we obtained. Here we can see that in the anti-A, anti-B and anti-AB, there is no reaction. Whereas in A1 cell, A2 cell and B cell, there is a 4 plus agglutination. In anti-D1 and anti-D2 also there is an agglutination, which is 4 plus. From that we can infer that the blood group of the sample we have given is ORH positive.
So thank you, Dr. Anand, for taking us and showing us how to type the D antigen via the tube technique. And also initially you showed us the typing using the slide technique and the later on moving on to the microplate technique. So we will be discussing the weekly testing in the next episode. And thereafter we will have a special episode and we will discuss all the question and answers, the practical tips and the various Viva questions which are asked for the D typing. Thank you.